All right. Good morning, everyone. Just going to give Zoom a few seconds here to filter everybody in, and then we'll get started here shortly. All right. Hopefully everybody's in. Thank you all for joining us for today's webinar. My name is Michelle Bertholet, and I'm the group publisher of agriculture here at Annex Business Media, which is Top Crop Manager's parent media company. First, I wanted to say thank you to Bayer Crop Science for sponsoring today's webinar and bringing us together to discuss a very important topic. I'm joined today by Dr. Charles Geddes with AFC and Rory Cranston from Bayer Crop Science, who will be giving a Western Canadian herbicide resistance update. Before I turn it over, I have a couple housekeeping items to note. This webinar is being recorded and it will be made available to all registrants and attendees approximately 24 hours after our live broadcast. This session is scheduled to run for approximately 60 minutes, including questions. And if you have any questions throughout the presentation, please feel free to type them into the Q&A tab found on the bottom of your computer screen. This webinar has been approved for 1 CCA CEU in integrated pest management, and further instruction for submission via QR code will be shared after today's question and answer period, where you'll be able to scan the code and self-submit for credits. The webinar also qualifies for certified crop science consultant credits. And if you wish to collect those, please submit a request through the CCSC self-reporting database at ccsctracking.ca, and I will share that in the chat later, or we'll share it in the email that goes out after the webinar as well. And without further ado, I will turn it over to you, Charles and Rory.
Okay, all right, thank you very much. So again, my name is Charles Geddes, and I'll be presenting first here today, and we'll, both Rory and I will be giving a little bit of an update on Western Canadian herbicide, or on herbicide resistance in Western Canada. So to start off, I'm a research scientist with Agriculture and Agri-Food Canada. I work in the area of weed ecology and cropping systems based out of Lethbridge, Alberta. And our research program focuses fairly broadly on herbicide resistance, where we look at discovery of new types of herbicide resistance, where they occur across the prairie region, and then develop strategies to manage those issues as well. So today I'm going to cover a few different topics here. I'll start off with a fairly broad sort of mention of herbicide resistance in Canada and the status of resistance in recent years, and then we'll look at herbicide resistance mechanisms. So we're going to get a bit more in depth on what causes herbicide resistance. I'll be providing a few examples with different mechanisms of resistance, and then we can talk a little bit about strategies or solutions to help. And I know Rory will be getting into that here after I'm presenting as well. So to jump right into it here, this is a global map showing the number of unique herbicide-resistant weed biotypes by country. And when I say unique herbicide-resistant weed biotype, what I mean is the combination of a weed species resistant to a herbicide mode of action. So in Canada here, we're sitting in third place, you could say, for the greatest number of unique herbicide-resistant weeds, trailing behind the United States and then also Australia. So we're sitting in third place, and unfortunately, first place is not the place you want to be in this competition anyway. So when you look at the discovery of these different types of herbicide-resistant weeds over time in Canada, you'll see the timeline on this map goes to about 2021, so it does need to be updated for the last couple of years. I do know in our program we've added a few new ones to this list just in the last couple of years. So we're sitting at roughly 80 to, I would say, 83, somewhere in there across Canada. If you look at the timeline here in years, you'll see that the herbicide-resistant weeds have been discovered at a fairly steady pace since the mid-70s, currently averaging about 2 new biotypes being discovered across the country each year. And so increasing herbicide resistance is important when we consider the limited number of herbicide modes or sites of action that we have to manage these issues. So this figure here is just showing the discovery and release of new herbicide modes of action over time, represented by different group numbers, as you can see here. And essentially what I'm pointing out here is that there is a steady release of new herbicide modes of action over time, which almost ceased in the early 80s. And so you can see there's been one group that has been released here recently and currently is of limited use in our cropping systems in Western Canada. So the point here is that we're seeing greater impact of herbicide resistance, but the herbicides that we have to manage these weeds are limited based on their activity that they have on plant species. And so this is just an example. This is a poster that I made for just outlining the different herbicide-resistant weed biotypes that farmers are dealing with here in the prairie region of Canada. So you can see they're organized by their group number. So all of these have been confirmed over the years. And we also organize the group numbers by roughly where they're having activity in the plant, whether it's light activation of reactive oxygen species, impacting cellular metabolism, or impact on cell division and growth. So you can see largely the greatest number of herbicide-resistant weed biotypes that we deal with are those where herbicides are impacting cellular metabolism. And here we have all of the different group 2 resistant weeds. You can see a large number of broadleaf weed species that have been found with group 2 resistance in the prairie region. So now I wanted to jump into how weeds evolve resistance. And so this is something that I think we don't communicate enough when we talk about herbicide resistance. But in fact, there's multiple different mechanisms or ways that weeds can evolve resistance to herbicides. And this is just broadly showing an outline of different mechanisms that can confer resistance to herbicides. So you can see that they're organized broadly into 2 different groups. The first is target site resistance, here shown by TSR, and then the second is non-target site resistance. And so basically target site resistance is something impacting where the herbicide actually binds in the plant, but then non-target site mechanisms are those that impact the herbicide before it even reaches the target site. And so what we're going to focus on here is kind of an upper level, and I'll provide different examples of these different mechanisms that can confer herbicide resistance. So we'll start off with target site mutations. So these are by far the most common mechanism to confer herbicide resistance in weed species. And so what it ends up being is you have a herbicide active ingredient here shown in green, and then the enzyme target site or the protein target site that the active ingredient would bind to in the plant. So your active ingredient in your herbicide would bind to this target site, and generally the active ingredient would inhibit part of a pathway within the plant that would ultimately result in breaking that pathway and causing the plant to die. And so here, when we have a target site mutation, the target site is actually altered, right? So basically, the target site is encoded by DNA within the plant, and you can have a mutation in, say, a nucleotide that results in an amino acid substitution that changes the conformation of, or changes the way that the target site can bind to different products. And so when your active ingredient tries to bind to that target site, it has reduced binding or no binding, depending. which renders the herbicide less effective or ineffective. And so an example of that, this is a recent case that we've been working on in Western Canada. An agronomist contacted us suspecting that they were dealing with group 1 or ACCase inhibitor resistant foxtail barley. And so I'll just kind of talk through the process here that we use to confirm resistance and then also confirm the mechanism of resistance. So you can see here, this is just a photo of the dose response experiment. First, when we would receive a population with suspected resistance, and this is similar for most of the diagnostic labs across the prairies as well, what they would do is a whole plant bioassay where they would grow, plant the population in a greenhouse or a growth chamber, let it grow, and they're planting it along with a susceptible sample as well. They would just treat it with a field rate of the herbicide and then see how it responds compared with the susceptible control. The next step then is to go on to a dose response experiment, which is really the research portion where our lab comes into play. And so this is a dose response experiment, or one replicate of a dose response experiment, where we have 5 different populations. You have PR1 to PR3. These are putative resistant foxtail barley samples that were submitted to our lab. And then we have 2 susceptible controls that we're comparing to in this particular dose response experiment for quizalofop. And so the field rate kind of being roughly in this area. And you can see that the 2 resistant populations are surviving the field rate of quizalofop in this case. And so these populations came in from the Peace lowland ecoregion, or the Peace River area of Alberta in 2022. And so here, just pulling out some of the data from this particular experiment. This is the quizalofop dose response based on biomass dry weight 21 days after treatment. And it's as a % of the untreated. So the blue and pink lines here represent the susceptible controls, and then the other colors represent the putative resistant populations. And so what we found overall, when we confirm resistance, the standard way to confirm resistance is to fit a dose response curve, pull out the dose of the herbicide that causes a 50% decline, in this case in biomass, or 50% management of each of the different samples that we're working on. And then we look at whether there's a difference between the putative resistant and then the susceptible controls. And so what we found here, this is showing the R to S ratio, or the ratio of the dose that causes a 50% response in the resistant to the dose that causes 50% response in the susceptibles. And so what we found is that roughly 2 of the populations were resistant to quizalofop, and they survived between 50 and 29-fold the rate of the herbicide compared with our susceptible controls. So we confirmed that these 2 of the populations were in fact resistant to quizalofop. But when we look at a similar dose response here with clethodim now, which is another group 1 herbicide, but from a different chemical family, what we found was actually there was no difference in response at the field rate. You can see at very low rates, you can sometimes see a stimulation of growth, which is actually tends to be quite common in herbicide research at low doses, where you can have this stimulation effect. But if you're looking at the field rates here, we do see, or we don't actually see, a difference between our resistant and our susceptibles. So the next step from there is we know that 2 of the samples are resistant to quizalofop. And so what we did was we extracted DNA from resistant and susceptible plants. So we have 3 resistant plants on the top over here, and then 3 resistants on the bottom over here. And so this is just a partial nucleotide sequence alignment for the ACCase, or part of the ACCase gene. And so if you look between these 2 vertical lines, what we're looking for is a base change in the resistant versus the susceptibles. So the different colored peaks are just calls for different nucleotides. But if you look at the center peak here for the susceptibles and compare it to the resistance, you can see that there's actually a bit of a difference there. For the resistance, we actually had 2 calls for different nucleotides. And so what this is suggesting is that there is a there was a mutation in this part of the ACCase gene. And so aligning it, what we found was that it was an isoleucine to asparagine, or it resulted in this amino acid substitution at position 2041 in the ACCase gene, so a target site mutation that is known to confer ACCase inhibitor resistance. We then went a little bit further and developed a rapid test where we can actually separate, we can test plants based on leaf tissue, extract the DNA from leaf tissue, and look at that DNA. And here we're just showing different groupings of the wild type or the susceptible plants here versus the resistant plants here. And what we found was that our resistant plants were heterozygous for this particular mutation conferring group 1 resistance. So that was an example of a target site mutation. But if we move on to increased expression of target site genes, this is another potential mechanism to confer target site resistance. And so I'll focus here on gene duplication, just to roughly outline how this works. So again, back to our active ingredient here and our target site. When we have gene duplication, there is some form of crossing in plants that ends up resulting in multiple copies of target sites. And so with further crossing, you can see the number of target sites ends up increasing, and you're applying your field rate of your herbicide. It's binding to some of those, but it's not filling all of the target sites, meaning that the pathway is continuing on in the plant, either unobstructed or in this case, less obstructed. And so the example here that I have for gene duplication is glyphosate resistant downy brome. So this was a sample that was submitted to our lab in 2021. And so it was submitted from an agronomist near Tabor County. You can see here the dose response experiment with glyphosate. Field rates are roughly in this 450 to 900-ish range. And our resistant sample in the back survived well above the field rates. From our dose response experiment, we found that the resistant sample was surviving 10 to 12-fold the rate of glyphosate compared with 2 susceptible controls. So here just showing a map of where Tabor County is located in the prairies. But then we went through a similar process to what I was explaining for the foxtail barley, but we didn't find a target site mutation. So then the next step was to look at potential gene duplication. And what we did find was that the target site for glyphosate here, represented by EPSPS, was 29-fold greater EPSPS copies within those resistant downy brome plants, suggesting that there are multiple target sites for glyphosate in this case. And to fill all of them, you would have to apply well above the field rates. And this is similar also to the mechanism of glyphosate resistance in kochia. And so this is showing essentially the same thing. If you focus on subfigure B here, we're looking at the EPSPS to ALS ratio. ALS is actually the target site for group 2 herbicides, and then EPSPS, again, is the target site for glyphosate. And what we're showing here, these are kochia populations from Montana, but the idea is that the ratio of EPSPS to ALS suggests how many more target sites you have for EPSPS. So we're seeing in those plants that have a higher EPSPS copy number, we're seeing it survive a higher rate of glyphosate. Here, looking at the LD50 value or the dose of the herbicide that is resulting in 50% plant death of these samples. So I'm going to skip over reduced absorption. We'll just explain that briefly, but this is usually related to foliar applied herbicides, and it is conferred by some alteration, generally some alteration morphology of the plant leaf that results in reduced uptake of the herbicide. So I don't have an example to show you in this presentation just for sake of time, but we'll talk about reduced translocation, which is often looked at in tandem with reduced absorption. So for reduced translocation or vacuolar sequestration, here we have in the target site, and this blue circle around the active ingredient is just representing, say, the active ingredient being moved into a vacuole within the plant cell, which reduces its ability to move to the herbicide target site. So it's reducing the amount of herbicide that's actually making it to the target site in this case. And so an example of this is reduced translocation of dicamba in kochia. So this is one of the known mechanisms of dicamba resistance in kochia. And I say one because we know that there are multiple. This particular example is some research that was looking at a susceptible kochia population or susceptible kochia plants here on the bottom compared with a dicamba resistant kochia plants here on the top. And so what they do in this particular study is they would treat a single leaf of the plant with a known rate of radiocarbon labeled dicamba. And so the treated leaf here is shown just by the red arrow. And the different graphs or the different figures as subfigures as we're going from left to right here is just different time points in hours after treatment. So if we focus in on the susceptible kochia here, you can see this leaf was treated, but the imaging shows that the dicamba actually translocated well throughout the plant. And you can see this in the subsequent images as well. But if you look at this dicamba resistant plant, it was treated on this particular leaf, so we would expect that to show up, but the translocation or the movement of the herbicide within the plant is impaired in this case. So when we pull out some data here, so this is data from the same experiment, focusing in on subfigure A here. This is looking at overall translocation with the resistant plants here in with the filled in black circle, whereas the susceptibles are the white circles up here. And essentially what we're showing is the percent of absorbed dicamba that was moved within the plant. And you can see there is much less movement of the dicamba in the resistant plant versus the susceptible plants. Looking at the other subfigures here, ATL is above the treated leaf, TL would be the treated leaf, and then BTL is below the treated leaf. And so what we would expect is, if the plant was susceptible, is movement of that dicamba throughout the plant, but especially above the treated leaf to where there's new growth and development here. And you can see with the resistant population, there's much less movement of that dicamba above the treated leaf. Most of the dicamba is actually staying within the treated leaf in the resistant population. And then there's also, depending on the time, there's some movement, but less below the treated leaf as well. So the last mechanism that I'll talk about here is enhanced metabolism, and this is certainly a concerning one for many weed scientists. And so essentially what happens here is selection for plants that have an increased metabolism for the herbicide. So the active ingredient, when it's applied, it's taken up, and then it's rapidly degraded into less active metabolites within the plant. So it's being detoxified within the plant before it even reaches the herbicide target site. And so here I'm just pulling out a few generalizations between target site and specifically this would be target site mutations versus metabolism-based resistance. And this is, there are different implications for each of them. So for target site resistance, it's usually a very, it's a random, naturally occurring and very infrequent mutation that occurs in plant species that happens to confer resistance to a particular herbicide. So it starts off, say, 1 in 100 million plants have this mutation. The herbicide is then applied, and the surviving plant is the one that is resistant. So that plant goes to seed, the seed enters the seed bank, and if over time, as you're applying the same or a similar herbicide, you see this selection happening, where your seed bank is slowly transitioning over to more resistant individuals and less susceptible individuals. So it's generally selected for by recommended doses of the herbicide or lethal doses of the herbicide. It confers a high level resistance, usually at the plant scale anyway. And there's lower potential for cross resistance because the mutation is specific to the herbicide binding site or the target site. And in this case, herbicide rotation or mixtures with different modes of action could help mitigate selection for resistance. For metabolism-based resistance, however, it often happens a bit more slowly, where recurring application of sublethal rates or low-level rates of herbicides in a field or particular areas of fields select for individuals that have a higher rate of metabolism or can detoxify those low levels more quickly. So as you see recurrent applications of sublethal doses, you're selecting for plants with higher metabolism. The issue, though, is that it more readily confers cross resistance to other products or other modes of action. And so this is the big concerning part, because you can select for metabolism-based resistance with one particular herbicide, and this could confer resistance to a different herbicide or even a mode of action that hasn't been discovered or released on the market yet. So that's why it's quite concerning for weed scientists, and should be for farmers and agronomists as well. And so in this case, herbicide rotation is potentially a less effective strategy to mitigate this type of resistance. And so just to pull out some examples here. So this is some research that was done by Hugh Becky on different wild oat populations from the Canadian prairies, here just named after the town or that they were closest to. And the different columns here are different active or different classes of active ingredients. So our ACCase inhibitors would be group 1 herbicides, ALS would be group 2 herbicides, and then different chemical families within that. So named differently here because this was a scientific publication, but this would be the FOPs, the DIMs, and the DENs. And so the different, a darker red here means that a higher percentage of resistance within the population. But when they were looking at the different mechanisms that confer resistance in these different wild oat populations, they found multiple different target site mutations that would confer resistance to group 1, as well as a substitution here that would confer resistance to group 2. But you also see enhanced metabolism in these populations, in this case conferring sort of a more broad level cross resistance to group 1 and 2 herbicides. And in some cases, this happens in tandem with target site mutations as well. So this is why herbicide resistance in wild oat is so complicated. And so here, these are some old data from 2001, but it's looking at different samples of wild oats collected from different fields, tested with 3 different active ingredients within the FOPs, and then 3 within the DIMs of group 1 herbicides. And you can see that there certainly is variable cross resistance to these different active ingredients, even within the same mode of action. And so over here, I'll pull out clethodim as an example. So this particular study did not find clethodim resistance, but we do find clethodim resistance on the prairies. However, it does seem to be at a lower frequency than some of the other group 1 active ingredients. And so this information can be used and implemented by farmers, especially when selecting herbicides for weed control. This is just pulling out the information from the crop protection guide, or the blue book in Alberta. There are similar guides offered in Saskatchewan and Manitoba as well. But on page 45, at least in the Alberta one, you can see that there are different chemical families listed under group 1, where we have the FOPs, the DIMs, and the DENs, and you can see the different active ingredients or the different products on the market that are within each of these different chemical families. So this can kind of help inform some of your herbicide choices. So one of the projects that I have the pleasure of leading are the prairie herbicide resistant weed surveys. And so this is showing some information from the previous round that was led by Hugh Becky before I joined AFC. So this was done in 2014 to 2017. The survey covers about 800 randomly selected fields across the prairie provinces. And in the background here, we're showing the percentage of fields over time as these surveys have been conducted, the percentage of fields with at least one herbicide resistant weed present. And this is increasing fairly steadily in all three prairie provinces, sitting between roughly about the high 50s to the low 70s as far as percentage of fields that have resistance. And so these surveys are integrated with rough mapping of the weed patches within fields, but then also a grower management questionnaire that asks questions about the perceived cost of herbicide resistance. And so we can aggregate this information together to come up with the cost of herbicide resistance for farmers. Here, just showing the data from 2014 to 2017 suggests that about 35% of field area under annual crop production is infested with herbicide resistant weeds, equating to about 59% based on field area. The perceived cost of resistance by farmers across the prairies averaged at $13 per acre. And this equates to roughly $530 million a year for farmers across the prairies. And so here, I'm just getting into some of the more recent data. So we surveyed Saskatchewan in 2019 and 2020. Manitoba was done in 2022, and then Alberta in 2023. And so it takes a while to get all of the resistance data and screen through all of these samples. So I'm going to touch on here the survey of Saskatchewan very broadly, where we can pull out various levels of information, but the percentage of fields that were tested that had herbicide resistance was sitting at 74%. This equates to an estimated 72% of all fields under annual crop production across the prairies. Just over half of those fields were occupied by herbicide resistant weeds, or half of the area of the fields on average, and then the acreage that's infested with herbicide resistance in the province. So we estimated herbicide resistance just for Saskatchewan farmers costs about $340 million a year. And so wild oat is one of the most common herbicide-resistant weeds that we have on the prairies. Here we're showing Group 1 herbicide resistance. The map is on Saskatchewan, but the graph here is showing across prairie provinces and over time. This is the percentage of fields where wild oat was tested that were confirmed with Group 1 resistance. And here, the survey of Saskatchewan suggested that that about 77% of the fields where wild oat is tested were Group 1 resistant. Group 2 resistance, we actually, at least in this Saskatchewan survey, we didn't find an increase in Group 2 resistance compared with data from 2014 and 2015. But you can see about 30% of the fields where wild oat was tested had Group 2 resistance. And then here we're combining both Group 1 and 2 resistance together. Looking over here, we're sitting at about 26% of the fields where wild oat was tested had both Group 1 and 2 resistance present in the same field. So this, of course, is an increasing issue because it drastically limits the herbicides that are available to control wild oat. And so we also know that wild oat can be trialate resistant as well, and we're working on gathering those data or updated data on trialate resistance across the prairies as well. I wanted to touch here a little bit on kochia now. And so here we're looking at the data from the surveys across the prairies. So kochia is actually sampled post-harvest compared with the other weeds that I was talking about, like wild oat, for example, which are sampled pre-harvest. So the kochia surveys covered roughly 300 populations in Manitoba in 2018, rough similar number in Saskatchewan in 2019, and then also Alberta in 2021. And so here we're looking at aggregated data across the prairies, specifically looking at glyphosate resistance. And we have it separated into low, moderate, and high resistance based on the percentage of individuals in the population that exhibited the glyphosate resistance trait. So low level resistance is something that may not be entirely obvious at this point, because when herbicides are registered for control of weeds, the threshold is 80% control. So 20% uncontrolled completely can be expected when applying herbicides. But so that's why low level resistance may not be completely obvious, but with continued selection, it increases into moderate and high level resistance. So what we found overall was roughly 3/4 of the samples, kochia samples tested across the prairies, had some level of glyphosate resistance, and the majority of those were either moderate or high level resistance. Here now looking at dicamba resistance across the prairies, you can see it tends to be more abundant in the mixed grassland and moist mixed grassland ecoregions, or the brown and the dark brown soil zone on the prairies. And overall, we found that just under 1/4 of the samples tested had some level of dicamba resistance, but almost all of those had low level resistance. So this is indicative of a problem that is coming down the road. And so here we're just looking now, combining the results for glyphosate and dicamba, and showing that overall 78% of the populations had either glyphosate or dicamba resistance, and 20% had both. So just to touch a little bit now on the story of herbicide resistance in kochia. So I'm going to use Alberta as an example because this is where we have the most data. And so here you'll see ALSR, or acetolactate synthase inhibitor resistance, which is group 2 herbicides. And so group 2 herbicides, or kochia has been resistant to these herbicides for quite some time. The first biotypes were found in 1988 for group 2 resistance, and now all kochia populations tested are group 2 resistant. So that's why it's at 100%. Glyphosate resistance in kochia in Canada was first documented in Warner County, just sort of southeast of Lethbridge here. And a survey the subsequent year in 2012 documented 4% of populations with glyphosate resistance at the time. 5 years later, that increased to 50% of those populations had glyphosate resistance, and 18% had dicamba resistance, which was not present in the 2012 survey. And then we fast forward again to 2021, and this is showing kind of a rough 10-ish year time frame, where we're now sitting at 78% of populations in Alberta with glyphosate resistance, 28% with dicamba resistance, and then 44% with fluroxypyr resistance as well. Just due to the way the project was designed, we looked at fluroxypyr resistance only in the province of Alberta. So fluroxypyr and dicamba are both auxin mimics, or group 4 products, whereas glyphosate is a different mode of action. And so some other interesting information that was pulled out of these surveys, because in Alberta we've done now 3 surveys over the years for herbicide resistance in kochia, you can see how resistance is changing. So this is looking at resistance incidence, or the percentage of individuals in the population that have that resistance trait. And over time, you see this distribution shifting further and further to the right. And what this tells us is that not only are we finding glyphosate resistance in more populations that are sampled in the province, but also it's having a greater impact in those populations where it does occur as well, because there's a higher incidence of resistance. The other thing that was interesting from these surveys was the overlap, or lack thereof, in resistance to dicamba and fluroxypyr among kochia populations. So we found overall 44% of the populations had some level of fluroxypyr resistance, 28% had some level of dicamba resistance, but only 14% of populations overlapped and were resistant to both. So what this tells us is that it's actually more likely that you have resistance to dicamba or fluroxypyr than you would have resistance to both of these auxin mimics. So potentially different mechanisms that are conferring resistance to these different active ingredients. And so this is the last thing that I'll focus on here today. This is the new type of resistance in kochia, where we're looking at group 14 resistance. So based on the surveys we've already covered, that all kochia has some level of group 2 resistance. About 3/4 have glyphosate resistance on the prairies. About 1/4 have dicamba resistance. And then in Alberta, we found 44% with fluroxypyr resistance. In the States, they also have group 5 or photosystem 2 inhibitor resistance, which we do not know to occur here, but we haven't been screening for it either. And now the 5th mode of action to which kochia has been found resistant is the PPO inhibitors, or group 14 active ingredients. So the story here, this was a population or a sample that was submitted to our lab in 2021 from West Central Saskatchewan. And we found that the population survived the field rate of saflufenicil. And so we conducted our dose response experiment comparing the resistant to 2 susceptible controls. And what we found was that overall, roughly about 80-fold resistance to saflufenicil applied foliar. And so this is just a photo of the dose response experiment. There was a single plant in each pot. This is just one replicate overall with the resistant in the back, 2 susceptibles in the front, and a varying rate of saflufenicil. 25 is sort of a rough field rate that we used in this particular experiment. And you can see that the resistant population certainly is surviving that field rate, and even surviving 10 times that field rate. Here's an image of a similar dose response, but now instead of saflufenicil, we're looking at carfentrazone. So rough field rates kind of highlighted here. And we actually survived up to 100 times that field rate of carfentrazone. So we've done preliminary dose response experiments with various group 14 active ingredients that are available on the market. And this isn't all of them, but it's a good number of them. And so this is some preliminary work that we actually have the proposal in right now to fund this research, but we had to get a head start to get information out to growers. So what we're finding here, we have different group 14 active ingredients and the resistant to susceptible ratios for this first susceptible population and this is second susceptible population, showing that we found resistance to saflufenicil, carfentrazone, tiafenicil, pyroflufen ethyl, all applied foliar. But interestingly, aceflurofin, it was actually susceptible. So aceflurofin comes from the diphenyl ether family of group 14 active ingredients. And so they appear to remain effective on this group 14 resistant kochia, but the unfortunate fact is that this particular family of group 14s is used less on the Canadian prairies and tends to be used more in soybean production. And then as well over here, we're looking at soil applied dose response experiments, one with flumioxazin in a sandy loam and a clay loam soil, where we found resistance. And then sulfentrazone, I also have to be determined here. And so this is just showing the photo of the initial dose responses with saflufenicil in the clay loam soil and the field rate highlighted here in red, where we did, the population is resistant to sulfentrazone applied, soil applied sulfentrazone. But even the lowest rate provided complete control of our susceptible populations. So our dose response model wouldn't fit properly, but we do know that it is resistant to sulfentrazone as well. And so just to finish things off here, we went back and screened through all of the kochia populations from the prairie surveys between 2018 and 2021 and did not find saflufenicil in any of the 882 populations that we screened. So we only have one known group 14 resistant kochia population on the prairies to date, which is from West Central Saskatchewan. However, since this, in 2022, colleagues from North Dakota State University have also confirmed group 14 resistance in multiple populations in North Dakota as well. So we know that it is beyond a single field, and based on the history of herbicide resistance in kochia, we also know that that this type of or that resistance tends to spread quite rapidly based on the biology of the plant. So we can expect that likely about 10 years down the road, group 14 resistance could be widespread on the prairies. And so in summary, I mean, we're giving this presentation on herbicide resistance because we know resistance is a growing issue in Western Canada. And so understanding what causes resistance and the issues that you have in your fields can help inform the development of management strategies. And there really is no silver bullet, but effective tools are available to manage these issues. And so with that, we will pass it off to Rory. I just want to thank first all of the various funders that support our research on herbicide resistance. This research would not be possible without their support. And as well, all of this research happens. It's really a team that makes this happen. So without all of these individuals here, this really would not be possible. So thanks very much for listening, and I'll pass it off to Rory.
Perfect, Charles. Thank you very much. So some great information there. I'll start sharing my screen here right away. Now, I'm going to probably try and go a little bit quick here. I definitely do want to make sure that we are leaving time for as many questions as possible. There was some great ones that have already come into the chat. There are some great ones that were actually on the registration that have come into that we definitely want to take some time and address as well too. So, but I do want to talk a little bit about solutions from Bayer, some innovation that we've got coming down the pipeline, and really concentrate on some exciting things that we have here, as well as some things that maybe I'd like us to start thinking maybe a little bit differently about going forward to really combat herbicide resistance. The one thing I've kept saying now is weeds and herbicide resistance are going to outsmart habits, but they're not going to outsmart systems. So really trying to go and focus on that. But first off, I do want to talk a little bit about innovation, bringing things to a farmer's field and to your sheds there, of what it actually takes to bring a new active ingredient, mode of action into into the market. So it really is a long way. This is what Bayer's pipeline looks like on development. There may be different names, but I'm sure our competitors do have something that look, fairly similar like it. And it's really interesting because if, we're talking PPO resistant glyphosate or PPO resistant kochia, but really we're talking group 2, group 4, group 9, group 14 resistant kochia. And if Bayer said today we're starting a project that we want to have something that controls all of these, it's about 10 to 14 years till something like that can actually go through all the regulatory hurdles, all the screening to actually develop something along that line. So it really is quite a long time. And really it's about a $400 to $500 million process for one AI to come through all of this. So definitely it's a lot of money and innovation that goes into it that people within the industry, ourselves and our competitors are investing into innovation. The one thing I will say too is through a lot of these phases, for something to get all the way through all the phases, we call something of a 1% success rate wildly successful. Historically, really about only 1 in 100,000 substances reach the market. So if you think one AI costs $400 to $500 million to commercialize, what are all those other ones we're investing in, having to kick out along the way? Now, the one thing that is nice about this, and it's probably the same for a lot in the industry, is this is changing just with new technologies. In the past, a lot of the times it was designing compounds or building them out, trying them in a greenhouse, and it was a bit of a shotgun approach of seeing how they work, what's going on, maybe if we tweak this and this one. But with a lot of technological advancements, this is getting better. So what I'm actually using as kind of an analogy is things like 23andMe or Ancestry.ca, where we send off a saliva sample and we can come back with information of, what are you genetically predisposed to? Where is your heritage from? Well, the nice thing is we're able to port that technology into a lot of other areas now. that we can actually start sequencing a lot of these weed DNAs and genomes too. And Charles showed a few there of we can actually 3D model and starting to use AI supported molecular target to really identify those structures and then build compounds so that we're not maybe having that 1 in 100,000 that, hey, we can get a really close before we actually start trying these things into the greenhouses, into the field there, that we're getting a lot closer there. So this is rather exciting, I think, for the industry that hopefully we can speed up things. We can be a little bit more laser focused, working with a scalpel versus that kind of shotgun approach there. So I'd say that's really exciting, I think, for Bayer and the industry of being able to use this artificial intelligence to really drive innovation going forward. I do like to show this. Bayer really is focused on innovation, and it's going to lead into my next slide there that when we came together as two companies, Heritage Monsanto, Heritage Bayer, we combined our innovation budgets to really drive out those next steps, those next products. But in total honesty and transparency, the $2.8 billion, it is the biggest in the industry, but that just doesn't include herbicide, fungicide, seed treatment development. It's also including breeding as well too. So Bayer really is committed to that innovation. You see 10.1% of our revenue goes into innovation, into that R&D, building it forward there. And this is where we're able to come up with the next generation of things here. And I've shown this around. This is a part of our investor deck here of we do have things coming down the pipeline, something really exciting. And for Western Canada is really going to fit in well for some of our driver weeds, some of our most prevalent weeds in the prairies, that we do have a new herbicide molecule coming down the pipeline. It really is exciting. If we think about some of our driver weeds, wild oats, we think about some of the other grasses that we have in there, that what resistances are we fighting today? Group 1, Group 2, and as we're seeing on here, Group 9 resistance. This molecule, this AI, is going to be a very effective tool for controlling in the future. Now, this is definitely looking really far out into the future. As this is currently in phase 3, we're going through all our rate titrations and really trying to understand it. But it is kind of exciting that we are coming along to be used in various market segments, not just one, that we could be using it in several of our crop rotations out here. So this is definitely exciting coming down the pipeline. Now, the question that I know is going to get asked in chat somewhere is, hey, what mode of action is this? And the answer is it's under classification right now. It's in with HRAC, and it goes through there. But the thing is that I can tell you is think of weed symptomology of Group 1s, Group 2s. We kind of have maybe that yellowing, chlorosis, or interreddening, or Group 4s or Group 9s. The differences there. Or even into the Group 6s or something quicker of just how this looks. Forget all this. This is going to be completely new symptomology, something we've never seen before. So rather exciting from a Bayer approach here. I will say, I think we'll be a little bit tight on time, but I think both Charles and I have, we've committed to staying on a little bit after, answer a lot of those questions. But I do want to talk a little about our newest product here that, once a product's registered, our innovation doesn't stop. We do go back to the pool here to really improve what we have. And so we can use Varo and Varo FX as an example. So Varo registered in 2008, formulation oil dispersion. Really any other formulation, in the market, suspension concentrate, wettable granules. But we have heard of, for Varo, Velocity, Bayer, those ones, so that, for improvement of efficacy. And it's interesting because I saw in the questions there that, will surfactants help maybe in some of these different herbicide resistance actions? And really surfactants are to get that AI into the plant quicker. So it doesn't say, isolated on the top. So it's really focused on getting it into the plant. Now, how much they help when they're in their plants, I'm not sure of that. Potentially a surfactant company could help us on that. But notes for viral velocities, we say add AMS or NIS when there's the 3 L's, what I call it, lots of weeds, large weeds, or low night temperatures to really help with that efficacy. And really, other than the oil dispersion, suspension concentrates, wettable granules, no other formulation was actually possible with this one. And this is where Viral FX comes in, is it's going to be really the first new Group 2 EC formulation. And, that doesn't sound that exciting, but it actually means quite a bit here for just improvement of efficacy as well as ease of handling as well, too. And you can see it just in the notes for Viral FX improvements of efficacy, as there isn't any notes for it. EC formulation really reduces that reliance on external adjuvants, but also it increases, and I'll show some pictures of here, the ease of use, greatly improvement handling, that you're not going and having to do as many rinses. It's not going to be hanging up in your sprayer as much, but also true bulk stability. I'm sure lots of people love smashing, 40-acre jugs into their chem handler on repeat, but having a 320-acre drum for your Varo FX is really going to hopefully improve that ease of handling and getting things in and through your sprayer as well, too. Just a few things that we do have here from the marketing side that I will talk a little bit on. Going to still be very flexible on that recopying aspect. No Group 2 hang up or carryover into next year. Improved handling, mixing for growers, easier tank clean out, more consistent grass control with that formulation. I'm going to hop in here just to a little bit of the results, just to show them here. So going to talk wild oats, driver weed, and the prairies, of course. So just Varo to Varo FX, 91.6, 95.8. So 4 or 5% doesn't seem like a lot of extra control, but just from formulation alone, that's the difference there. That Floxapure we know is going to be doing nothing on our wild oats, but this is just going from TCM or thiocarbazole methyl in an oil dispersion and to TCM in an EC formulation, which is truly exciting. Talking about kochia, since that is a driver weed, and Charles did touch on it a little bit there, but just showing that kind of advancement there, Varo, we kind of assume that Group 2 kochia is just everywhere. I say don't ever bank on your Group 2s working, but showing what that FX Floxapure in Varo FX does bring, and then the tank mix partners as well too. So Varo FX plus Infinity or 240 MCPA where they are. But even that bump up of Varo FX to Buctril M, involving that bromoxynol, that Group 6 in there, I think to really help out that herbicide resistance of those Group 4s as well too, is very valuable. I did want to touch on for sure the ease of handling of Varo FX. This is one thing I think really excited about, but just making those passes a little bit more effective in our field. This is something that came out of our trials this year. So top 2 pictures are Varo in Chem Handler. If you've ever used this product before, you've probably experienced this. Oil dispersion, adding it to water. Well, we know how well sometimes those mix, but so we have Varo on the top left corner before being rinsed and right after being rinsed in the Chem Handler. We always say it's good to have more water, more rinses and getting in there, and it is very evident with this one. We have seen this before, but we always kind of, when I look at this, the thing that pings in my head is with any Group 2 is tie up within the sprayer, potential contamination into your next crop. How well is that? That's happening here. What's happening in all those little nooks and crannies in there? On the bottom left, we see Varo FX, so that new formulation in the Chem Handler there. Nice, there's still a little bit of residue in there, but after that first rinse, definitely some good looking there, so not worried about tie up there. And the next thing I do want to talk about is a little bit, and this is going to be brand agnostic. I'm going to use of course, a Bayer variety, a DeKalb variety. But one thing I think we need to get a little bit more comfortable with is using Roundup Ready and Liberty Link or TrueFlex Liberty Link stacks. I know there's that hesitation with a lot of people of, oh, it could be a super weed in a lot of cases. And I think it's really easy to control volunteers, and we're not developing anything more in here. And why I actually think that we need to get a little bit more comfortable with this is we showed some of the maps and I kind of pilfered them from earlier surveys, but if we think of kochia and wild oats, and really we when we look at those surveys, we kind of concentrate on our weed now. And the one thing I like to actually am thinking now and driving out more is what if we start drawing Venn diagrams of the fields that have, you know, glyphosate resistant kochia on it, as well as group 1 resistant wild oats. What do we do on those fields? And this is where I think that, you know, having flexibility of what you can choose for your systems there to really do, or do both. So if you got group 1 resistant wild oats on your field and you're growing Liberty Link variety, you know, that clethodim may or may not do a lot of work on it, as we kind of saw from some of the information Charles had earlier. But I'm really afraid to burn that out if we're relying on that. And we know that Liberty maybe doesn't have the greatest activity on wild oats. Is that where the glyphosate or Roundup comes in? And then, you know, vice versa for glyphosate resistant kochia and just a solo true flex crop, that if you've got both in the field, this might be the solution for you to really kind of push a lot of the weed control out of there. So really kind of get the best thing there. So I really do kind of want to see it, get a little bit more comfort with those, as those varieties, their performance is getting higher or equal to kind of what's on the yield in the market there. I think they're going to provide a lot of flexibility on what we can be applying to our crops and there. So I do think that maybe some people are concerned that we're going to be using too much glyphosate or Roundup on our wild oats, but to be honest, I'd like to challenge that a little bit on how much Roundup are we actually applying to our wild oats. I think we think that we apply it pre-harvest, we apply it pre-seed, we apply it in crop. That's 3 application on wild oats. And to be completely honest, I'd like to challenge a few of those. Of that pre-harvest one, often seeds have set already and it's not going to affect it. Pre-seed sometimes, I think with a lot of the agronomy that we have done in the past is we're seeding earlier and earlier and earlier and earlier. And we know wild oats are a later emerging weed. So I think a lot of the times we're in the field or we've sprayed that before they're even emerged. So I'd actually, you know, I know Hugh Becky has said that this is the potential for the next glyphosate resistant crop, and I agree with it if we're putting the same application pressures on it. But I truly do wonder how much we are putting on for application pressure going forward. So I do think there are some potentials there for using this or utilizing this for those fields that have multiple resistances on it, or just as kind of something to control that flexibility, change systems up. Because as I mentioned earlier, herbicide resistance figures out habits, but if you put systems in and you keep it guessing and you got a flexible system, it's only going to be a benefit for you. So that's a lot there. I have included the CCA, CCSC QR code there as well too. I think we're a little bit over time, but I'm definitely committed to staying on for a while for any questions that have come up. I believe that there are several in the chat that we should definitely discuss. So.
Wonderful. Thank you very much, Rory and Charles. That was great. Yeah, let's dive into some questions. You can scan the QR code on the screen there if you're interested in collecting the CCA credit for integrated pest management. And you just hold your phone up to the monitor so the camera can hover over the code and it should open up your CCA app for you. All right, so we have lots of questions. So we'll try and do a handful here, and then the other ones we'll maybe get back to the attendees via email, or if I can convince Rory and Charles to answer some of them via video or text, we'll try and get that up on the website in the next week as well, and share that with registrants, because there's a lot of conversation happening here. So let's start with one that came through the chat. Folks are interested where they could find the weed survey publications. And Charles, when is the next round of surveys scheduled to begin?
All right, that's an easy one. So thanks for that. So yeah, the Weed Survey Series publications are published by Agriculture and Ag-Food Canada, and I mean, the easiest way would be to email myself, and I can send you a PDF of past publications. But also, I'm happy to announce that we have a new project that's funded, that funds the next round of surveys across the prairies, starting with Saskatchewan again in 2024 and 2025. Manitoba is scheduled for 2026, and then Alberta for 2027. And so there'll be a publication on each of those provincial surveys, but also we're working on developing a Prairie Weeds website where all of this information would be openly available to whoever is interested in looking at the status of herbicide resistance. So that's under development, and should be available here within the next year anyway.
Wonderful, thank you. All right, there were several questions, both in registration and I think one here in the Q&A. Can you walk us through what you recommend growers and agronomists do if they suspect resistance, and where can they send those to be tested?
Sure. I assume that's directed at myself. So if a grower suspects that they have herbicide resistance, it's recommended that they submit a sample for testing to confirm whether it is resistant or not, because in addition to confirming that the issue may in fact be resistance, the testing can also help confirm whether it's due to other factors like weather, or issues with either application or other factors that contribute to lack of control, right? So I would say about at least half of the samples that come in actually do not end up being resistant, at least the ones that come into our lab for a research perspective. Our lab offers free testing for herbicide-resistant weed biotypes that are not known to occur across broad acreage. So this would not include things like glyphosate-resistant kochia, group 1-2 resistant wild oat, but we're focused more on the novel resistance and new biotypes, and doing the research to provide that baseline of information to other diagnostic labs that can then offer tests as a service for farmers. So there's labs across the prairies and elsewhere even that offer this as a service for farmers. So most of them are whole plant, based on whole plant bioassays on the prairies, as I sort of touched on a little bit in the presentation, which work well to confirm resistance regardless of the mechanism that is conferring resistance. But we're also working on developing rapid genetic tests that will be able to take leaf tissue from an uncontrolled weed in the field, extract DNA, and look for the mechanism of resistance. And with that information, we'll be able to tell with fairly good certainty which herbicide active ingredients the specific weed is resistant to. So we're working on developing those. There's one available on the prairies through Pest Surveillance Initiative in Manitoba for glyphosate resistance in kochia already, but we have a list of 12 or 13 more that we've developed over the last few years.
Perfect.
Yeah, if I could just add in, the whole thing is I say get it in, either to Charles's lab, or even if you're not happy with the product and you get it in through your chem rep. A lot of the times we get these submitted off to Charles's lab. We had an experience definitely with a resistant sample this year between mine and Charles's lab that came in through us, and we're going to be in complete agreement getting it to them and sharing as much information as we can. And to be honest, the benefit there is a lot of the times samples will go to Charles's lab, but then we'll also send them off to Germany too, that we can start building that next iteration of an AI to come along the pipeline. So whether you send it in yourself or it's sent in through your chem rep, be it whatever company, just send it into the proper lab, or even through your chem rep. There's lots of venues and avenues to get it in.
Perfect. Rory, let's jump to Jeremy's question here. Do you expect that even with the new technologies, we'll be able to outpace the herbicide resistance in our no-till systems And is that the best investment, chemical development direction for battling this challenge?
Well, the interesting thing here, and if you want my honest opinion, Charles is probably laughing, but I look at our last mode of action that was brought in, which was the HPBDs in 1987. So even if we've got a new mode of action by 2030, that's going to be 40 years. So if you're asking me if we can outpace it of just chemistries, I don't think so. I think it's a losing battle there. We're on a treadmill that's speeding up as we go. That's where I do think that we need to really diversify, start looking at things a little bit more of a lot of the truly integrated weed management strategies there of how can we use different herbicides at different times if we're just looking at chemistries, but also, can we do some more spot controls of some of these patches or patch control? We've got some really interesting things coming down the pipeline within the industry of kind of that we'll call precision application or precision control of doing those. I think a lot of even looking into crops in general, that as we improve varieties, that competition, what we were able to do earlier with seeding rates and what we have for machinery is going to improve that too. So do I think if it's just chemistry and we change nothing else, we're going to fall off that treadmill, but we need to be looking at everything all in. So we're going to be definitely investing still in chemistry, but I think it needs to be investment in as many things as possible between digital, between even if we have to do min-till in some of those areas and patch control for sure. But that's my honest opinion. I'm not sure, Charles, if you've got any additions there.
Yeah, I think Rory, you hit the nail on the head there, right? It's really, I mean, I've talked about this issue quite a bit, and there's a whole body of research that we do that I wasn't able to present today on integrated weed management and integrating non-chemical weed control tools with the products that we have available to manage herbicide-resistant weeds. And time again, it shows that integrating these non-chemical tools, for example, like increasing seeding rates, decreasing row spacings, competitive crops, and so on, they actually interact with herbicide efficacy and even do help manage some of these resistant weed biotypes, even if the product you're applying, that specific population has some level of resistance to that product. These non-chemical tools help improve that efficacy. So it's really a combined approach.
Mm-hmm.
Yes, for sure. All right, we've got a couple more here that we'll try and get to. Danielle asks, does the time of application of herbicides impact the plant's ability to build resistance towards the herbicide?
Yes.
Yeah. So I'll maybe take a bit of a crack, but I even just think, well, the way I see it is, how I'm going to equate it to the human body, that it's going to take a lot more poison to take down me than someone half my size, right? So controlling a smaller weed is easier for sure, to actually get that out there. But if you wait longer and apply it and you don't get that lethal dose to it, bigger plants are going to have more target sites and you can't get as much on there, right? So that's how I view it. So Charles?
Yeah. So I mean, the size of the plant and the growth stage certainly does impact, I think, selection for herbicide resistance, right? Larger plants tend to need more herbicide to control them in the first place, right? And I mean, kochia is a prime example. Generally, once it gets past 10 centimeters in height, most of the products that are registered for kochia control will not provide complete control. Right. So it's really stage dependent. And if you're applying to those later stage weeds, then the unfortunate consequence is that you're applying sublethal rates and potentially selecting for things like metabolism-based resistance, right? So stage certainly does have an impact. Another example could be with dicamba resistance in kochia. There's some research. Dicamba is quite often applied as a pre-emergence herbicide, but then also can be applied post-emergence at lower rates as well, right? We do see resistance to foliar applied dicamba, but in some cases there's been research showing that soil applied, or the residual activity from soil applied dicamba, still has efficacy on those dicamba resistant kochia. Cases, right? So that sort of difference in you're applying the herbicide also does have an impact because metabolites are changing in these plants as they're emerging and developing, right? So the stage certainly does play a role, and that's why there's a recommended window for application of any given product.
Great, great, thank you both. Let's maybe attack one more here. Rory, is there one in the Q&A that you'd like to finish up with?
Whatever you feel. There's lots of good questions in there.
There's
yeah.
lots. Let's maybe would a stacked trait be risky to select for resistance against the group 9 and 10? And how would you manage volunteer canola?
That's a really great question, and I could see how we get to that question for sure. So I can understand it, but if you put it into a system, no. We're looking at other traits definitely throughout the world that it's usually the resistance come and then the herbicide trait comes after that we need the flexibility to for a lot of those things. So if you're doing stack traits on repeat, that's where it comes into an issue. So if you're doing canola, snow, canola, snow, canola, snow, yeah, that's a lot of pressure that way. But if you're putting your cereals and your pulses around it, there's definitely a lot of great options. As far as how do you manage volunteer canola, there's actually a lot of great options out there that if you're controlling any other broadleaf weed, there's a good chance volunteer canola comes with it. You know, the one thing that we are pushing is mix as much as you can when you can. So I know one thing we are going to be really pushing is, you know, most Roundup, 70% of it on your pre-burns is going down alone. If we could probably change one thing, I would love to have more tank mix partners in there. And that's a very easy way to control volunteers with a lot of the groups that we have in there prior to our cereals, prior to our pulses. Any group 2 is going to be absolutely nuclear on your volunteer canola. So your in crops, if you're using a Clearfield pulse or you're doing a group 2 in your cereal, is going to be doing really good. Any group 4, any group 6 is going to be just great on canola. So there's a lot of volunteer canola management possibilities there. So do I think that it's risky to select for resistance to group 9, group 10? There's always potential there, let's face it. But it's the flexibility. Just because you have group 9, group 10 doesn't mean you have to apply both. You can see what weeds are in your field and adjust to it. It's more about that flexibility issue. If you say dual stack and hey, the kochia didn't take this year, but it's the wild oats, well, just maybe do the glyphosate, or maybe it's the kochia that took on and the wild oats were under control. Well, then you have the Liberty Link as an option. It really is about that flexibility to really adjust and go on the fly and not staying into the habit of one or the other. I don't know if you got anything to add to that, Charles.
I think you covered it quite well. We can probably just leave it there.
Okay.
Great, thank you both. All right, so that was, the final question for today. I thank everybody for staying online with us well past the hour. I'll make sure to share the questions with the presenters, and they can reach out to folks. But we'll also maybe talk about how we can put together some more resources and share it with all the registrants, because there was some really great discussion and requests for more information, right? So, all right. So with that, all the registrants should see a follow-up email in their inboxes within 24 hours with our contact information and the on-demand version of the webinar for you to watch. And again, thank you very much for tuning in to today's webinar. Thank you very much to our presenters and to Bayer Crop Science for sponsoring today's webinar. And don't forget to visit topcropmanager.com/webinars to view this recorded webinars and to register for any upcoming ones. Thanks again and have a great rest of your day.